Okay. Hello, everyone. Welcome. Thank you for signing up for our webinar. So my name is Gurinder Vinner. I'm a material scientist here at Dolomite Microfluidics, and I will be hosting this webinar today. So let's go to it. Okay. So the topic today we want to discuss is microfluidic generation of double emulsions for fluorescence-activated cell sorting processing. And before we do begin, just a couple of housekeeping items. If you do have any questions, feel free to leave them in our questions chat box. It should be on your screen, and we will address those at the end of the presentation. And if we don't have time, we will email you those as well. So just to keep that in mind. So if you have any questions during the presentation, please feel free to leave those on there. Okay, so today I hope to tell you a little bit about our Dolomite particle engineering team, what we do here, and how we use double emulsions for FACS, introduce our microencapsulation system, which is what we use to make these double emulsions, and then also how these double emulsions are made using that system. And I will be able to give you a live demo of doing that. Once I've shown you that, I'll just close off with some concluding remarks, and then finally I'll take and answer some questions if time permits, or otherwise I will email them to you. So our Dolomite particle engineering team, we are a team of chemists and engineers who focus highly on high performance nanoparticles and microparticles with precision control over size, shape, and structure. We use microfluidics in flow chemistry as well, but we also have a big focus on biomedical and pharmaceutical industries, and this is what this double emulsions for FACS it comes under. We also have some standard products that we provide, which are things like particles and emulsion stabilizers, and one of these emulsion stabilizers is also what I'm going to use today in our demo. And we also do other things such as advising customers on microfluidic setups and processes. So if anyone's interested in the use of microfluidics for their research, we certainly provide that kind of advice. And for other customers who are slightly unsure on how their chemistry or biology can be implemented with microfluidics, we also offer proof of concept or proof of principle projects for customers ahead of any equipment purchase, so they're definitely sure that it's something that's suitable for them. So what is microfluidic technology? Microfluidic technology is essentially something that can allow you to have an enhanced end product. With conventional techniques, you would simply mix two immiscible fluids, as you can see, a water and an oil, for example, and you would have a homogenizer which breaks this liquid into tiny droplets. But the result of something like this is quite poor in the sense that it's a very monodispersed population of droplets, and you have little control over size, loading, and therefore the downstream processing as well. And this also uses a lot of energy, and there's a lot of reagent waste as well. When you move on to using something like a microfluidic device to produce these droplets, you can immediately see that the product, the end product is much better in the sense that the droplets are precisely controlled. You can use less reagents to process your sample. You have higher throughput, and there's basically reduced cost. So this all applies even in biological applications where you are looking for a higher throughput and you want to use small sample volumes, which something like conventional techniques does not permit you to do. So this is essentially what microfluidic technology is allows you to have precise control over your experiments or over your assays, and be able to manipulate that downstream very easily. So what about double emulsions for FACS? How or why that is important? So some of you may already be familiar with FACS. For those that are not, I'll just go over this very briefly. Fluorescence-activated cell sorting is just a method for sorting a heterogeneous mixture of cells into 2 or more containers, as you can see here, and it all happens at 1 cell at a time. It specifically relies on cells which are tagged with let's say with fluorescent antibodies, and that is how you would do the sorting. So if there's a fluorescent signal, your cell would be sorted in, let's say, the positive, and if there isn't a fluorescence present in the droplet, it would be negative. So a key example of something like this is directed evolution. So with directed evolution, you can use proteins or antibodies. You can have a large gene library. The important thing here is that when you do randomization, mutations and things like that, you want to be able to screen a large library, which currently with normal assays you wouldn't be able to do. So this is where you would get a higher throughput if you were to use droplet microfluidics to sort those types of droplets. The outcome of this type of artificial evolution experiment is crucially dependent on 2 things. One is the number of variants that can be screened, and the second, that's the quality of the assessment of each clone that forms the basis for selection. So definitely for the first aspect of this, microfluidics and droplet microfluidics can come into play as it offers a higher throughput. So the size of the gene libraries that can be obtained from these experiments easily exceeds the throughput of any screening system, which currently wouldn't be possible. So implying that screening is the bottleneck in the exploration of sequence space. So again, once you have your droplets, you can either have your transcription, translation going on in the same droplet. These can then make a substrate fluorescent if the correct gene is present. And if that's the case, then you can immediately sort that through the FACS sorter into a collective population, which you can immediately downstream analyze, allowing you to have a genotype to a phenotype linkage. So a standard water in oil emulsion, so this is just a water in oil droplet, wouldn't be compatible with a FACS. And why that is because the FACS sheath fluid is also aqueous, so you wouldn't be able to have an oil phase in there. So that's why it would be incompatible. And that is why we move on to using double emulsion, so a water in oil and a water droplet. So these emulsions here, you can already see there's your cells and the enzyme are present in the aqueous droplet, then they are surrounded by an oil shell, and this is then immersed in a aqueous solution. So what is our microencapsulator system? So our microencapsulator system is a complete system for high throughput encapsulation of individual cells into picolitre droplets. So this enables you to work with very small volumes, which is essential when you've got a rare or a precious sample, and allows you to basically have multiple assays running one after the other. It allows you to encapsulate single cells, DNA, enzymes, antibodies, anything you wish, and allows, if you're working with, let's say, mammalian cells, it enables you to encapsulate up to 300,000 cells in approximately 15 minutes. There are 2 channels, so you can co-encapsulate. So one channel could introduce your single cells. The other channel could be your functionalized beads or a reaction mix, which get mixed into the same droplet and allow the reaction to occur downstream. So it's an ideal way of profiling natively paired T-cell receptors, isolating antibodies, coding sequences, encapsulating cells in hydrogels, or encapsulating expression libraries for FACS sorting. We also have our FCC, which allows a certain degree of automation and integrated droplet analysis with the PC software. So on your left-hand side, you can see this is the basic setup of a microencapsulator system, and I will also show you later on in this presentation. So the basic components of a microencapsulator system are our 3P pumps. So the 3P pumps are able to supply fluid. So one would be your two would be your aqueous channels. One would be, let's say, pushing your cells, and the other one would be pushing the reaction mix or your functionalized beads. And then the third pump would be your oil phase to capture those or pinch those droplets at the junction. So that hence why you would need 3 P pumps. These are then mounted with some flow sensors and interfaces. The flow sensors are designed to accurately detect flow rates between 1 to 50 microliters per minute, although we have other flow sensors if you wanted to go higher or below that. And then we have our temperature control unit here, which allows you to control temperature from 1 to 100 degrees, especially if your assay is sensitive, temperature sensitive. Above the TCU, we mount our microencapsulator module, and this is where our droplet junction chip and your reservoir chip, where you load your cells and leads, are loaded. So just a top view of the TCU and the microencapsulator module here. We've got the sample reservoir chip. So here's the reservoir chip. This is where your samples would be loaded. The volume of this is 100 microliters. So you would be able to load 100 microliters of your, let's say, bacterial cells and 100 microliters of your reaction mix. And then we have our 2 reagent droplet chip, which fits into this slot here. These do come in various different sizes, 50, 15, 30, or 50, depending on what is the final drop size that you desire. And again, this all can be controlled in terms of temperature, as it's directly in contact with our TCU. So even upon collection, which is here, the product remains under a controlled temperature. So what about double emulsions using a microencapsulator system? So this is what a typical process would look like. So let's say you have your bacteria coming from channel 1, and we are encapsulating 1 bacterial cell per droplet. And at the same time, this is being mixed in with the enzyme substrate. So these droplets contain a single cell. It could also be a mammalian cell, it doesn't have to be a bacterial cell. And these are then collected into a collection tube. Upon collection, the reaction here would occur. So if there's a fluorescent probe, this will initiate that reaction. And then this droplet is then reloaded back into another droplet chip, which now would be a hydrophilic chip. And that allows you to then re-encapsulate these primary emulsion, we call them, into another droplet. So then it becomes a water in oil water droplet. The purpose of the second channel here is to cause spacing in between the primary emulsion so that you don't end up encapsulating 2 primary droplets in one big droplet. And the resultant solution is something like this. So you have your aqueous drop with a shell of oil, and then all of that is immersed in some aqueous solution. The typical oil that we recommend using for any biological application, and it's biocompatible, is one of our products, which is FluoSurf, which is also readily available, and most of our work that we carry out here is usually done with FluoSurf, so it has excellent biocompatibility. So again, as just reiterating here, we use a 30 micron fluorophilic chip for the first stage, so when you're making the primary emulsion. And once you have done that, we have to switch the chip around to a hydrophilic chip. All that means is our chips have 2 different coatings. One is fluorophilic and one is hydrophilic. That essentially means it allows you to produce really stable water in oil emulsions when you use a fluorophilic chip. And when you use a hydrophilic chip, it allows you to make stable oil in water emulsion. So you do need to switch them around and simply produce those. When you do use a 30 micron chip, you can successfully make very monodispersed 20 to 30 micron droplets. That is not limited to that. If you can manipulate the flow rates as well as use the 50 micron or the 15 micron chip if you desire a slightly different size of droplets. So that's definitely possible. Once you've collected your droplets from here, you can use a FACS sorter, which is probably available on your site, and it will work with any standard FACS sorter. So whether you're using ARIA or Sony, these emulsions are compatible and could be used to sort with those. So just to move on a little bit to the demo right now, I'm going to show you how the P pumps look, what the P pumps look like, and I've got the system running at the moment. So typically I would have loaded cells in one of the channels, and I would have loaded beads in the other channel. So this is holding a volume of 100 microliters, and this is also holding a volume of 100 microliters. And this is where the continuous phase, which is the oil phase, which is going to pinch those droplets, is coming from the top and the bottom. And that's allowing the pinching to occur. And once the droplets have been made, they are then released via this channel and then are collected downstream into an Eppendorf tube. So I'm just going to pause here and take you through our system. And so just bear with me a second. All right. Okay, so hopefully you can still see this slide. Yeah, I think you can. So what we've got here is the system that I've just been showing you, our microencapsulator system. We've got the 3 P pumps, our pressure pumps. They are all connected via this tubing. We've got some inline valves in place, which allow me to manipulate the flow in real time. You can see that one of the P pumps has a digital display. This is our advanced pump, and these 2 are our basic pumps. Although they both do the same job, this just allows you to do something manually rather than use a software. So these then deliver the fluids which are placed into these containers here, and this is then connected to the TCU, and we are able to view in real time using our high-speed camera and our microscope. The way this is connected, because that's the experiment running right now, we have the microencapsulator module, which looks something like this when it's mounted on our TCU. And then, as I mentioned earlier, we've got our reservoir chip. So this is what a reservoir chip would look like. This would typically go in the middle here, and using a very standard laboratory pipette, you would load your sample with a P200, and then you would use your droplet chip. So the droplet chips are a little bit smaller, and they look something like this. This then gets slotted into a side here, like this, and then you simply close that and connect the tubing, which is then connected to the P pumps. So this is, again, not at the moment mounted on the TCU, so you are able to work without the TCU if you don't want temperature control. And then once the droplets have been produced, they are collected into a typical Eppendorf tube, and then you have a cover on top, which allows you to maintain some temperature if you desire that. So once we've got that, we've got an FCC running here. I will show you a little bit of a pre-recorded, just because this is live at the moment and it's difficult to share. But so you can see that the droplets are being produced. This is a 13 micron fluorophilic chip. And we've got our FluoSurf, which is an emulsion stabilizer, pinching the droplets from this way. And we've got, let's say, the cells and the beads coming through this way. And you can see very nice monodispersed droplets being produced. If I so desire, in real time, I can manipulate the size of these droplets by simply changing the flow rate. So by doing that, so you can see immediately the droplets are much, much smaller. So there's a lot of control over how you can manipulate your droplet size, and also throughput. So once you have some conditions in mind, you can always set everything up, and it's very, very reproducible. You're able to put in the same flow rates, and it'll give you the same size every single time. So that's what it looks like. In order for me to talk you through a little bit more about what our FCC, which is a flow control center software, looks like, I'll switch back to our presentation and talk you through how that works. Okay, so hopefully you can see my screen again. So this is what the FCC kind of looks like, as I've just shown you live. So you can see we've got 4 windows open here. One is obviously showing the live streaming, which is by a microscope and the high-speed camera. And then we've got a few other pop-ups, one being the FluoSurf, which is our continuous phase And I'm able to select the fluid calibration, so it's accurately showing me what flow rate it's going at. So I've set it up to 35 microliters per minute. I can work either in pressure mode or flow mode, but to get a little bit more reproducibility every single time, it's always a good idea to keep in flow mode. And the real-time graphs on the side also show you how stable your flow is over time. So if you do tend to go away from your workstation, you can always come back and check, if your flow has been disrupted or not. So that's always nice information to have at hand. So we are able to get log files if you so desire to figure those out. At the bottom two windows, we've got the cell channel and the buffer channel, which I'm controlling as well. And again, I can rename these pumps to allow myself to recognize them better. Again, I can select my fluid calibration, and I can set them at whichever flow rate I would like. So by doing this, I'm able to control real time how the droplet changes. So just to show you that a little bit better, you can see by changing the flow rate here from 35 to 15, I immediately change the size of the droplets. So again, you can see there's a real-time change in your droplet production. And I'll probably go back to 25, and you can see that changing. So it might take a couple of seconds just for it to get to stabilize a little bit, and then you're good to go. So by simply manipulating the fluid flow rate, you can dramatically control your droplet size. If you want to go a little bit higher or more, you can simply use a smaller or a bigger droplet chip. So just to say this one is a 30 micron chip, which means that the 30 microns is here. So the channel here is 30 microns. So next, to make the double emulsion, so to make the double emulsion, I would simply change the droplet chip here, and then I would load my primary emulsion into one of the channels. So this is the emulsion that we made in the previous slide, and then actually use some fluorescence, which is otherwise called the spacer fluid. So this will just space those droplets. And then my continuous phase now changes from the oil to aqueous, which is probably some between 20 you can use TBST. And again, I'm using a 30 micron hydrophilic chip. So this is what something like that would look like. You can see that I've got my primary emulsion coming through one of the channels, and I've got a spacer fluid, which is fluorescence, coming through the other channel. Again, I've relabeled my P pumps so I can understand them better. So this is my continuous phase TBST. I've changed my fluid calibration because it's aqueous now, so it can more accurately detect what flow rate this is going to be. So that's 7. And then I've also got my primary emulsion and spacer oil, which I'm controlling. And you can see over time, it's pretty stable. There's no apparent fluctuations that are of concern. And you can see, it might be difficult to tell if, depending on how well you can see there's double emulsions being made. So in every single droplet, there's a droplet inside. To show you this better, I have taken some images. So these are just some snapshots I took earlier. You can see this is the primary emulsion that we produce in the first stage. This is now coming in. Here's the spacer fluid. So it's sort of splitting the primary emulsion. And you can see that there's a very thin, nice oil shell, enough to keep the emulsion stable. So this is how it's being produced. And upon collection, you can view this very easily on a glass slide. So I've just collected them on a glass slide. And you can see there's a nice ring there. There's lots of double emulsions. On the odd occasion, you might see a single emulsion. What that basically means is when the primary drop hasn't been encapsulated and you just simply have the spacer fluid and the TBST making a drop. So that's what I mean by single emulsion. So this is typically what it looks like, and it's very monodispersed, and it's quite compatible with a standard FACS machine. So finally, when it comes to when you've collected your emulsion and you're ready to FACS sort, there are just a number of things we usually recommend. We are not essentially the experts. You know, some of you might be FACS expert, but we do say, you know, you need to calibrate your FACS machine, you can do this simply by using your calibration beads that are available to you by the provider. And then I would also recommend that before actually performing your actual experiment, is to collect some empty droplets, so an empty double emulsion, which is made just simply using water in oil and water without any of your pressures or rare reagents. And this will allow you to sort of gate according to the morphology that you see. So it is important that when you do make this empty emulsion, that you make it according to the same parameters, same flow rate as you would your actual experiment. So it allows you to just prime the system and use it as a reference point. Typically, 100 microliter sample should give you approximately 0.2 to 5 million droplets. Once you have collected this, we usually recommend that you just sort of dilute your sample in 1:5 ratio in a FACS diluent buffer. This could be 1% between 20. You can use others as well, whichever you prefer. And typically, if you are working with too smaller volumes, so this is anything below 15 microliters, be aware that you will need to have manual agitation during a run, as there will be some settling. So that's not ideal. And similarly, if it's too much, if there's too many droplets and this is exceeding 100 microliter sample, you will eventually start seeing breakage, and this is not, again, ideal, and you wouldn't be able to collect your final sample as effectively. So it's very important to collect the right amount and have enough so that you can successfully sort them. So a sorting event, if you notice that you're sorting something that's sorting event of more than 2,000 per second, this greatly signifies that there will be breakage going on, and you can either lower your pressure on the FACS, or you can abort your run completely. Ideally, you want to be aiming for something like just under 200 events per second, and again, the parameters for pressure will differ with each instrument. So with the ARRI and the Sony, there are some differences, but that's something that, again, you can work with an engineer from each of those instruments. But usually, once you have the parameters in place, they are quite similar in how they perform. So that's also worth noting. So those are just some of the key points that you want to keep in mind. Okay, so just to sum up what we've spoken about today, the microencapsulator system that I've showed you today, it enables you to produce double emulsions, which can easily be FACS sorted, and it can work with any standard FACS sorter. And the same platform can be used for your primary stage and the secondary stage. You just need to switch around your chips, which is very, very easy to do. We do have some user-friendly protocols and support available if you so desire. We have some protocols which I talk you through each step. So I've just talked to you about this very briefly today, but we do offer some insight into what kind of pressures to use and flow rates to use. Or if any other custom support you would like, we are also available at hand for that. So the microencapsulator system enables other applications as well. So if tomorrow you decide that you want to do some single cell analysis, so that will enable, this system will enable you to do that as well. So you can see single cell encapsulation, agarose, cell culture, stem cell analysis, and there's many more applications which you can perform using our microencapsulator system. If you do want to find out more, you feel free to get in touch or even browse our website. And then we have Dolomite products and particle engineer teams expertise, which enables users to achieve high performance and reproducibility. So again, help is at hand if you do so require. If you have any queries, please feel free to leave them. And finally, thank you for listening. I hope you have learned something today and it's been informative. If you do have any questions, I will check those now and we will reply. But thank you once again for attending.